Hey guys, so we're going to continue on with heredity stuff and with Punnett squares, but today we're looking at some kind of unique scenarios with heredity. So we're going to be looking at both incomplete dominance, codominance, as well as multiple alleles. So all of those are scenarios where you don't just have a dominant version of a trait and a recessive version of a trait. There's something else kind of going on. So we'll look at how to deal with those Punnett squares today. Okay, so like I said, we're looking at incomplete dominance, codominance, and multiple alleles today. Essentially, you need to know the difference between codominance and incomplete dominance and how they differ from like a normal scenario. Then you have to be able to do Punnett squares for both of those. Then you have to explain how multiple alleles can affect a trait and do Punnett squares or traits involving multiple alleles. And then we're just going to briefly do some definitions at the end. So incomplete dominance happens when you have two alleles for a trait, but neither trait is actually dominant over the other. So in this case, if a person has a heterozygous genotype, the phenotype will be a mixture of the two alleles because neither one is able to outcompete the other. And you'll end up with a kind of intermediate trait. So for example, in snapdragon flowers, the two alleles for flower color are red and white. So if an individual is homozygous for the trait, they will be either red or white. But if the individual is heterozygous, they'll be the mixture of red and white, pink. And then codominance happens when, once again, you have two alleles for a trait, but this time both of them are dominant. So both will be expressed just in different areas. So in this case, a heterozygous individual, so someone with both alleles, would have both traits fully expressed in different areas of their body. So for example, there are these type of flowers that could be either red or white. And so the homozygous versions will be either red or white. And then the heterozygous version is red and white. So with incomplete dominance, the heterozygous individual would be pink. With codominance, the heterozygous individual is red and white, just in like splotches. So when you are writing the genotypes for both incomplete dominant alleles and codominant alleles, you can either write each allele as its own letter, so like RR for red, WW for white, and RW for the mixture, the either red and white or the pink. Or sometimes you'll use like the same letter as like your base and have a little exponent that denotes the actual alleles. So you might have like C for color, and then like the exponent RR for red, the exponent WW for white, and the exponent RW for pink, or the red and white. Okay, I usually do like the first version just because it's less writing and I'm a little lazy. But you should be able to recognize it either way. Okay, so just to kind of drive home what the difference is between codominance and incomplete dominance, in terms of what they would look like as homozygous individuals, they would be either red or white for both incomplete dominance and codominance. But with incomplete dominance, the heterozygous individual would be pink. With codominance, it would be red and white. So the next two slides are just a couple practice questions on how we would do this. So yeah, watch me do one or two and then pause and try the rest on your own. Okay, so for the first question on the incomplete dominance Punnett squares, it says that we have a red snapdragon, which, as we talked about before, in order to be red, they must be big R, if it's incomplete dominance. And then white must be WW. Sorry, let me just focus this for a second. There we go. And so if we are going to do a Punnett square of this is just going to be a little 4x4 because we're only looking at one trait, which is the color of the snapdragon. And so we'll put big R across the top, big W across the side, and fill it in. And so the genotype ratio would be 0, 1, 0 for red, red and white, and white. And then the phenotype ratio would also be 0, 1, 0 for red, pink, and white. Again, genotypes are the actual alleles, so the letters, and phenotypes are the actual physical traits. But either way, in this case, it is a 0 to 1 to 0 ratio. 100% of the offspring would be pink. For question number 2, it says determine the phenotype and genotype ratio for a pink and red snapdragon. So our red is going to be RR, our pink will be RW, and when we fill this in, we will get 2 to 0 as both our phenotype and genotype ratio. So the phenotype ratio would look like that. The genotype would be red to pink to white. Again, this could also be simplified as a 1 to 1 to 0 ratio, but it would be 50% red and 50% pink. For question number 3, it says curliness of hair is incompletely dominant. Straight hair and curly hair are the incomplete dominant alleles, and a heterozygous individual would be wavy hair. So that means big S would be straight hair, CC would be curly, and CS is the heterozygous, and that would have the intermediate wavy trait. It wants to determine the phenotype and genotypes for the F1 and F2 generations if the parents are both homozygous for different hair types. So if the parents are homozygous for different hair types, one must be straight and one must be curly, which means all of the offspring have wavy hair. So what that means is for the F1 generation, which is the first generation of offspring, you have 100% wavy hair, and it would be a 0 to 1 to 0, both genotype and phenotype ratio. And then to find the F2 generation, we're going to take 2 of the individuals from the F1 generation and cross them. So 2 wavy-haired people. And we will fill in our Punnett square like so. That will give us a 1 to 2 to 1 ratio, both for genotype and for phenotype. Okay, so again, the F1 generation phenotype and genotype ratio is 0, 1, 0. The F2 generation is 1, 2, 1. By the way, the only time you will ever be asked to determine the genotype or phenotype ratio of an F2 generation is when the F1 generation are all the same as each other. That way you can pick any 2 individuals and cross them and it'll be the same answer. Okay. Next up, we're going to go to the codominant Punnett squares. So in this one, question number 1 says, in shorthorn cattle, the alleles for coat color are red and white, and they are codominant. It wants to know the genotype and phenotype ratios of a white cow and heterozygous bull. So our white cow is going to be WW, and our heterozygous bull will be a red and a W. And then we fill in our lovely chart here. And it wants to know the genotype and phenotype ratio. So it's going to be for red, 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 white, and white, white, it is going to be a 0 to 2 to 2 genotype ratio, also a 0 to 2 to 2 phenotype ratio, where we have no red cows, we have 2 red and white cows, and we have 2 white cows. Again, that could also be simplified to a 0 to 1 to 1 ratio. It then says, what are the chances of a white progeny? Well, it would be 50% white, or half of the individuals. And then for number 2, it says, in some species of chicken, chicken feather is determined by black and white codominant alleles. Heterozygous chicken are called checkered. What are the chances of 2 checkered chickens producing a black chicken? So if we have 2 checkered chickens, that means that they have both the black and white allele. So if we fill in our Punnett square, it's going to look like that. And then it wants to know, what are the chances of producing a black chicken? Well, it would be 1 in 4, or 25% will be black. You'll also have 20% white and 50% checkered. Okay, next up we have traits that are controlled by multiple alleles, which means that there's more than 2 alleles. So for example, in fruit flies, the eye color can actually be coded for by 4 possible alleles. Usually the most common version of the trait will be called the wild type, and then all other versions are called the non-wild type or the mutant type. There's also sometimes a hierarchy of dominance among the multiple alleles. So sometimes you'll have one that's like the most dominant and one that's the most recessive. Sometimes you'll have some that are co-dominant, stuff like that. The letters are often uppercase with subscripts used to denote the different alleles, or sometimes you will just use different letters. And I'll show you examples of kind of what both of those would look like. So in humans, blood type is one of the traits that we have that is determined by multiple alleles. So you can either have the A allele, the B allele, or the like I allele. So sometimes they're written as IA, IB, and then lowercase I. Other times they're just written as A, B, and O, which mean the same thing. The A and B are codominant, and the I or the O is recessive. And then they can result in the phenotypes A, B, AB, or O. We can get the AB because, again, they're codominant. Okay. So if you have both uppercase A's or an A and a lowercase I, that would result in the A phenotype. If you have both B's or a B and an I, then again, the B is dominant over the I, just like the A is dominant over the I. So those would all be B phenotypes. Obviously, AB results in an AB blood type. And if you don't have A or B, that's how you get the O. Since it's recessive, you have to have 2 recessive copies. As soon as you have a dominant version in there, you won't get the recessive traits showing through. So let's do some examples of what Punnett squares would look like with multiple alleles. Okay, so number 1 says determine the phenotypic and genotypic ratios of the progeny of a heterozygous type A and a heterozygous type B person. So in order to be heterozygous type A, they must be type A lowercase I, because the only other version of type A is the homozygous, where they have 2 capital A's. And then same for our type B man. If he's type B, could be capital B, but that would be homozygous B. So the only way to be heterozygous B is to be the B with the I. I would also accept if you put O's where I'm writing the I's. And then we're going to cross these over. So AI and BI gives us AB, AI, BI, and II as our 4 different genotypes. And so then for the genotype ratio, there are a couple different options. Again, the person could be like capital A, or AI, or we should be colons, sorry, or AB, or BB, or BI, or II. The only ones that we have are 1 AI, 1 AB, 1 BI, and 1 II. And then for the phenotype, we have fewer phenotypes because both of the first 2 genotypes gives us blood type A. Then we have type AB. The next 2 both give us type B, and the last one gives us O. So of these, we have 1 type A, 1 type AB, 1 type B, and 1 type O. So this is our type A, our type AB, our type B, and our type O. Okay, for question number 2, it says that we have a type AB woman with a type A man. So our woman is AB, our man is A something. We don't know if he's AA or AI. It then says that this man's father is type B. So his dad is type B something. Could have either been BB or BI. Now, this A would have had to come from somewhere, and it definitely didn't come from the dad, because if the dad had an A allele, he would have been type AB. So the A definitely came from this guy's mom, which means that whatever else is here would have had to come from his dad. Now, his dad definitely didn't give him a B, or else this guy would be type AB, and it says that he's type A. So the only other thing that his dad could give him that's not an A and would still leave him as type A is an I. So we kind of have to work backwards between the dad and the man to figure out what the man's genotype is. Then we can figure out what the chances of these guys' children being type B is. So we'll do our Punnett square, set up the woman as AB, the man is AI. And so we end up with one child that's type AB, 2 children that are type A, and 1 that is type B. So 1/4 or 25% are type B. Okay, so that's it for our Punnett squares. Again, there are going to be more practice questions coming out to you for all these different types of Punnett squares. But the last thing I want to do today is just cover a couple definitions. So the first is polygenic traits, which are traits that are determined by more than one gene, not just more than one allele, but more than one gene. Essentially, this is going to be much more complex than any type of thing that we're going to be doing this year, but it is important to know that they exist. Continuous traits are traits where the phenotypes gradually vary from one extreme to another. So instead of just being tall or short, there's a whole continuum of different heights that humans could possibly be. Continuous traits are a type of polygenic trait. They're controlled by multiple different genes, which is why there's so much variation in them. An example of that is things like height and skin color in humans. Polygenic traits will have a lot more variability because there's a lot more different combinations of those multiple genes and their alleles. In humans, many of our characteristics are determined by multiple genes, and so sometimes it's difficult to figure out which gene is really causing a trait and how it's being inherited, because you don't know if that trait is coming from gene A or gene B, that kind of stuff. The other issue is that a lot of our traits are also influenced by the environment. Traits that are influenced by both multiple genes and the environment are called multifactorial traits. This includes things like eye color, height, fingerprints, weight, heart health. A ton of different traits are influenced both by your environment, and it's sometimes your environment in utero, like with your fingerprints, and your different genes. And then these polygenic genes can also be codominant, incomplete dominant, and have multiple alleles. So it further complicates the ability to predict traits of offspring. So when we're doing these predictions, we're assuming that we're dealing with traits that only have one gene coding for them and that aren't influenced by the environment. You'll see that we use things like eye color a lot, and technically the genes for eye color are brown and blue. But obviously people have like green eyes and hazel eyes and stuff like that. And that's because the environment also plays a role into how our eye colors develop. So yeah, genes are a lot more complicated than we're going to assume that they are for a lot of our Punnett square calculations and stuff like that. But just be aware that they're more complicated, and then we'll kind of forget about it for our Punnett squares. Okay, that's it for today. Let me know if you have any questions about the Punnett squares that we did.